we do in fact live on a dormant planet. So according to some estimates, around 60% of the biomass on Earth is in a dormant state. And we know this from the bacterial biomass, for example, but we also know dormancy in the context of cancer cells that can lay latent in the tissue and cause cancer relapse. But we also know this from cryptobiosis in insects, or even the famous examples of tardigrade dissections. But dormancy is also prevalent in some animals whose embryos are able to temporarily arrest their embryonic development in face of adverse environmental conditions. And so common to all of these systems is that they are able to enter a temporary dormant state in the face of adverse environmental conditions. And so understanding the biology of dormancy allows us to understand the biology of a large proportion of life on Earth. And it prompts questions such as, how does an organism slow down? How does molecular processes, how do they shut off temporarily? And then what in this scenario become the new rules of homeostasis? And how does a cell or an organism decide then what to keep or trash? So what are the molecular mechanisms that promote stability to molecules that you cannot afford to lose? So they might be, for example, important for keeping this whole process reversible. So in order to answer some of these questions, we are using the African turquoise killifish, Nothobranchius furzeri, and this is a crash course in their biology. So these fish are native to the Southeast Africa, where about 3 months of the year they swim around in small ponds. However, for the remaining 9 months, these ponds are completely dried out, and as you can imagine, this is detrimental to the adult fish. And so this whole species survives by the fact that their embryos are able to enter a dormant state called diapause, where they arrest their embryonic development for months or even years. And they are subsequently able to exit this dormant state when the water returns, and then the whole life cycle continues. So I hope you agree with me that this is now an excellent system to study dormancy, because we can keep these fish in the lab, we have access to their embryos because of their external embryogenesis, and these embryos enter dormancy as a natural part of their life cycle. So if I now were able to awaken your interest in dormancy biology, and you're interested to understand how these dormant embryo are regulating the energy-intensive process of translation, how are you asking these keep or trash questions by profiling global complex behavior, and how you store protein complexes, and how we are trying to figure out the order in which development is reestablished and the dormancy process reversed using single embryo mass spectrometry, you can find me at poster 23.
Thank you.